looking at the properties of these two different genetically distinct cell types, the somatostatin and the PKC delta. We looked at their properties both at baseline and also their function in the modulation of pain. And then the work that we've been following up afterwards is looking at the circuit level. And I'll show you one of the examples of the efferent projections is from the PKC delta neurons to the zona incerta. And then from the input level, we focused on the parabrachial nucleus. And these are the stories that are most complete and that I chose to share as an example of how we tackle cells all the way to circuits and behaviors. So the very first question we had was a lot of different labs have been showing that the somatostatin and PKC delta neurons in the CEA are functionally distinct in many different behaviors that are amygdala dependent. So our very first question was, are these cells only genetically distinct, or is there anything electrophysiologically or morphologically that characterizes these cells? And in order to begin to address that, what we did is we used Cree mice for PKC delta or somatostatin. That was too fast and now it's distracting me. Sorry. Okay. We use Cree mice for PKC delta or somatostatin coupled with reporter mice, and that allows us to visualize the PKC delta and somatostatin neurons by the fluorescence protein. And then we also couple this with slice electrophysiology, and we add biosighting inside of the internal solution so that then we can morphologically reconstruct the neurons and look at their morphological properties. Okay, so what we see is Anisha did this beautiful characterization that you can see at her paper, but the two main distinguishing features are clearly appreciated both in these videos and here in these representative traces. And the bottom line is that in addition to these neurons being genetically distinct, they're also electrophysiologically and morphologically distinct. So we have that in any parameter that we could look at, all the somatostatin neurons are hyperexcitable compared to the PKC delta neurons. And morphologically, you can see here that pretty much all the somatostatin neurons are compact and have complex arborization of the processes, whereas the PKC delta neurons have longer processes that are way more less complex, so a lot less arborization. So this demonstrates that the neurons are not only genetically distinct, but they also have distinct, unique electrophysiology and morphological properties. Now, to evaluate the function of these neurons in the context of pain, we use a cough model of neuropathic pain. And what this involves is that we expose the sciatic nerve of the animal and we put a little plastic cuff around the sciatic nerve, and then we perform a battery of behavioral tests to be able to evaluate cold, heat, tactile, and pinch hypersensitivity. And this is just as a proof of concept that when the animals have cough, you see robust hypersensitivity in all the different modalities. And of course, the sham has no effect on the hypersensitivity, on the behavioral responses. So using this model, we started by looking at whether the neurons, the PKC delta and somatostatin neurons, were differentially recruited in the context of pain. We use ERK as a pain plasticity marker that I have shown as a grad student. Is not only a marker, but it's also functionally important. If we block ERK, inflammation-induced hypersensitivity is reduced. So we use this as a plasticity marker. And when we look at the colocalization with PKC delta or somatostatin in the context of nerve injury, what we see was very clear that a lot of the cells were colocalized with PKC delta with virtually no overlap with the somatostatin neurons. So these results suggested that there's cell type specificity in the injury-induced plasticity, and that the PKC delta neurons are the ones that are being recruited in the context of injury. We then follow this up with electrophysiology in acute slices, and when we patch the neurons that express PKC delta, what we see is that, consistent with the histology data, we observe increased excitability in the cough animals compared to sham, and this is expressed as an increased number of spikes, as you can see here in the rep traces, and the summary graph, an increased number of spikes in response to the polarizing current injection. So basically, all of this together was pointing to the central, to the PKC delta neurons being involved in promoting pain. And in the next set of experiments, then we wanted to establish causality. Is this related to the behavioral hypersensitivity? In order to do that, we used a chemogenetic approach. So in the PKC delta Cree animals, we injected an inhibitory dread that will allow us to inhibit the neurons that express PKC delta locally only in the CEA. And then we performed a battery of behavioral tests. When we inhibit the PKC delta neurons, what we see, and this is just to orient everyone before I show the results, these are the PO withdrawal threshold. Lower values is more pain, higher values means less pain. And before the inhibition of the PKC delta neurons, the sham animals have less pain, and all the cough animals have hypersensitivity. And then when we inhibit the PKC delta expressing neurons, we see a complete reversal of hypersensitivity in animals that were treated with CNO. No effect on baseline hypersensitivity, demonstrating that the modulation of these cells, that the modulation of hypersensitivity is specific to injury, to injured states, and not just baseline nociception. Then we wanted to know if, in addition to the neurons being necessary for pain, would they be sufficient to drive hypersensitivity? So we did similar experiments, but now we injected an excitatory dread into the CEA of PKC delta neurons. And what we see is, again, less hypersensitivity hypersensitivity in the cough compared with the control animals. But now what we see is that when we activate the neurons, all the control animals that are sham or naive, so they have no injury whatsoever, now they show hypersensitivity that resembles the hypersensitivity observed after injury, but occurs in the absence of any injury. So this demonstrates that the PKC delta neurons can do both, increase and decrease pain, depending on whether we activate or inhibit them. So then we wanted to know what's going on with the somatostatin neurons. They have been shown by many different groups to be really important for amygdala-dependent behaviors, and our histology showed no colocalization with the pain activity, with the pain plasticity marker. So we turn to electrophysiology, and when we look at the activity of these neurons in acute slices, what we see is, first of all, that just like we saw in the naive animals, a lot of the somatostatin neurons in control conditions are spontaneously active. But then when we look at the neurons in the context of injury, we see that most of the neurons are silent at rest. So these results demonstrate that the activity of the somatostatin expressing neurons is decreased in the context of injury, which is the complete opposite to what we see in the PKC delta neurons, that we see increases in excitability in the context of pain. So we did similar experiments now to look at the functional significance of the somatostatin, or the functional contribution of the somatostatin neurons. And when we inhibit the somatostatin neurons, we see hypersensitivity in control animals that have no injury, demonstrating that inhibition of these cells is sufficient to drive hypersensitivity. And just as a parallel example, it's completely opposite to what we see when we inhibit the PKC delta neurons, when we see reversal of hypersensitivity. Now, when we activate the somatostatin neurons, we get this very potent analgesia. So the animals that are that have injury or the cough, they now they go back to even higher values than at baseline. And again, just as a reference, is the opposite to what we see when we activate the PKC delta neurons, that we see hypersensitivity in control animals. So based on these combined findings, we concluded that activity of the somatostatin neurons drives analgesia, and activity of the PKC delta neurons drives hyperalgesia, which demonstrates that the central amygdala indeed has this dual function in the modulation of pain, and that the effects are cell type specific. So this is just a cartoon of what we have found. We postulate that the central amygdala functions as this pain rheostat that can drive pain up and down, with the mechanism being cell type specific, so that when PKC delta neurons have increases in excitability and drive hypersensitivity, whereas the somatostatin neurons have decreases in excitability in the context of injury, and their activity drives analgesia. And this was really exciting because, as I told you, when I started in the field, I've always been puzzled about these two functions of the CEA, and we feel that we were able to demonstrate that both of those are modulated in the CEA, and we further provided a mechanism by which this is occurring. So for following up, now that we know that the central amygdala can do both, and now that we know that the central amygdala does this in a cell type specific manner, then the focus of the lab has been in identifying the circuit level ways that this bidirectional modulation is occurring. And in the first example, I'll show you studies that we did to look at the efferent pathways from the PKC delta neurons. To do that, we injected a Cre-dependent anterograde tracer in the central amygdala of PKC delta Cre animals. What this will do is that all the cells that express PKC delta will pick up the tracer, then it will be anterogradely transported to everywhere in the brain. And we looked at areas in the brain, the whole brain, that had terminals in the that were coming from the CEA. These very, very labor-intensive analysis revealed 17 different brain structures that receive inputs from PKC delta expressing neurons, and they're everywhere in the brain, basically, except the cortex. And this is consistent with the classical classical studies that use traditional neuroanatomical marker tracers that have demonstrated that the central amygdala projects down and never back to the cortex. And we can see, we saw some regions that had dense all the way to sparse labeling in all of these different structures. One of the structures that got our attention was the zona incerta. This is a ventral thalamic structure. And the reason that one got our attention from everyone else was because previous work from Asaf Keller's lab at University of Maryland, they had shown that in the context of pain, neurons in the zona incerta show decreases in excitability. And we know from our studies that the PKC delta neurons, which are the ones that go to the zona incerta,